Yeah, good evening, all of you. 5 students only. Okay, my is only joined. We start. Today's is of our lab session. One of our theory topic is antibiotics. And another topic is enzymes, fermentation, industrial applications of fermentation. These are all remaining topics. Antibodies means white blood cells. More importance takes care about for the formation and generation of antibodies. Okay. Okay. Antibiotics is the great scientist. Is in very earliest of this 19th century, means on 1928, Wilhelm Fleming This great scientist is identified as one of the antibiotic, the penicillin, is the product of fungus. Antibacterial properties also is a product of the fungus. Once is identified, after that is named as penicillin. The fungus molds. He separates from the laboratory. He is identified that this substance acts as one of the antibiotic substance. This entire thing and process is in 1940s. It's totally completed with the bulk. The product of penicillin is developed with the great the faculty members of Oxford University. He is there pioneered about this Harvard, the Florey and Ernest Chen. These both are forward a step in 1938. Initiate one of the German scientists and the one is the Australian microbiologist, German chemist. These both are in the ASA, penicillin, and industrial way of product is they generate in a bulk quantity. And at the same time, the simultaneous Second World War, a lot of humanity soldiers used to call it diseases. These people are also saved by survival to this the medicine. Yeah, is in a penicillins or cephalosporins. These are the antibiotics derived from the fungus. You know the fungus or cephophytes, especially. Sustainability of the fungus is in dead animals. It develops. Antibiotics can also derive from the other bacteria, not only fungus, other bacteria also. These are aminoglycosides and carbiphenins, aminoglycosides and carbiphenins is extract from the bacteria. And synthetically it's made up and so many modifications of the chemical assay and chemical structure, whatever the naturally occurred antibiotics. These all antibiotics act as antibodies, like it protects the disease. So that is the reason why first we learn about the antibiotics. The entire the role of antibiotic is save the life. It interrupts the disease, the body of the plant or animal or creature is destroyed. The malfunction takes place. The chemical synthesized antibiotics is totally full-fledged laboratory made. It's human made. It's anthropogenic one. All these sulfonamides and quinones, these totally synthesized. This is the era, the golden era of 1950s to 1960s. Why? Because a lot of antibiotics is identified. Sulfonamides is initially up to after penicillin. Once it detects next, it begins each and every scientist is they identify and separates the particular the bacteria or fungus cells in naturally growing one to synthesized and develops the antibody. just like how it is in a clinical trials on living animals like rats, rabbits, all these. Yeah, of course, sulfonamides, penicillin, streptomycin, chlorophenicol, tetracycline, erythromycin, vancomycin, ampicillin, methicillin, and cephalosporinsis. The recent fast of 2000 to 2005, these two, 2005 is in a daptomycin and lenzoid. These all are next after 1960s onwards, the new inventions of antibiotics. It's slow down. It's totally just only is a conventional way of some of the modifications. Onwards, developments. This particular the new antibiotics have been introduced, but there are modifications, conventional way of their change, but whatever the existed one is a natural one or else synthesized one. The sophistication techniques is developed, so sophisticated machines is also. So that is the reason why the easiest way of developers. Now new medicine era is begins through nanomedicine, nanopowders, nanocrystals through this particular any disease is identified and give the medicine. And next in between the nuclear medicines also, whatever the radioactive substance in a doped with the smallest quantities, very teeny particle of nano like the smallest quantities is doped, then the disease is escalate, acts as antibiotics, especially in carcinogenic diseases, cancer diseases. Yeah, like yeah, it's resistance. What it resists, it develops within a short span of time. So in a long, not in a long run of this particular antibiotic usage, but it can resist within a short span of time. Just like I said, meticillin. Meticillin is in a 2 years after the meticillin is introduced. It can resist to Staphylococcus aureus. One of the bacterium was identified. In case of penicillin, what it can, the resistance was identified even before the antibiotics was released to the widespread of usage of all it was not a problem to until antibiotics is began to used of intensively. Penicillin of Streptococcus. Yeah, from 1940s to this particular the medicine is non-stop working. You see how it is the great scientist of Alexander Fleming is identified. Is in laboratory conditions. Under the laboratory conditions, is separate that particular species. I think. The beginning days, the technology beginning days, or science beginning days, a lot of tactics of the human being, the food, shelter, and the disease point of view. So no technology, but at the same time, the great scientists are initiated. So all the things is escalate, surmount the people's diseases. The, the population is increases, and no sufficient food is not. Each and every country it's the same thing is they identify. And in between the wars, First World War, Second World War, is these days is very, very hectic days. So normal people, soldiers, and at the same time, the scientific community also they faced a lot. See, yeah, this microorganism, this is caused one. Disease causing substances. Yeah, next is clinical trials. Is a pet dish, is a cultured, high media agar pets. Yeah, this is the penicillin fungi. Penicillin is develops. These molds too he extract. clinical trials, mice or white rabbits. Types. Sword movement. Meningitis patient. This is the story behind penicillin. That particular the great scientist he mentioned. Whenever he identified penicillin. Then onwards, antibiotic levels, diseases, controls. Antibiotics. These all antibiotics in several classifications we will identify. What is antibiotic? In what way it's classified? Several ways of classification. How to uses and misuse or overuse. It's nothing but of overuse and the resistance and the cross resistance. Possible side effects, characteristics of ideal antibiotic is how it is the character wise. The antibiotic is in a naturally occurred one in a household. It's all ginger, some other plant leaves, it's garlic, it's lemon. There are so many. These are all are in the household. You can cheap and readily available in natural. But someone is the synthetic or synthetic type of agents, naturally occurred one and synthesized. But these are all the functionality is in suppress the growth of organisms, diseased caused one. It inhibits the growth, particularly microorganisms point. The semi-synthetic one is the chemically altered of existing natural antibiotic compounds. And synthetic one is full-fledged in a chemical compounds is synthesized. And is one to another is combines, is designs. Into another is in a combines means nothing but designs in labs, in the laboratory conditions. After that, once its assay is forms next to the clinical trials. The clinical trials, especially in vertebrates. The vertebrates is best one is simply available is mice or white rabbits. These are all our mammals. The mammal functionalities, everything these can identify. These are all are classified as the mode of action, the activity of that particular, the base of mode of action wise, activity wise, action wise, and the mechanism of action. The way of classification is identified. The microorganisms destroys through their mechanisms is protein synthesis inhibitors, cell wall synthesized inhibitors, nucleotides, DNA nucleotides containing of this particular DNA synthesized inhibitors, RNA synthesis inhibitors, folic acid inhibitors, and mycolic acid synthesis inhibitors. Their mechanism of antibiotics is classified in this several ways. Yes, these are all which one is comes under penicillin and cephalosporins, vancomycin, yeah, and beta lactamase inhibitors. These and one is the cell wall synthesis inhibitors, and another one is polymyxin and bacitracin. Bacitracin is bacitracin. These comes under beta lactamase inhibitors. And on the other hand, the rest of one of the RNA synthesis inhibitors, DNA synthesis inhibitors, is the majority of the fluoroquinolones and the metronidazole. These both are comes under DNA synthetic inhibitors, and RNA synthetic inhibitors is reformed. Isoniazide is comes under mycolic acid synthetic inhibitor, and the folic acid synthetic inhibitors is sulfonamides and trimethoprim. Yes. And protein synthesized ones is it inhibits 30s and 60s size of the proteins. Why? Because these are all are amino acids. Most important of 20 amino acids already we are discussed. These amino acid synthesized one is in aminoglycerolide and tetraacylene, or else in microlates and chlorophenicol and clindamycin and streptogamines. These are all inhibitors of protein synthesized one. According to their mechanisms, is a major role. Next, this particular the broad spectrum of their activity. The activity based is the tetracyclines and chlorophenicol, amoxicillin, and cephalosporin, and erythromycin. These comes under this particular antibiotics classification of their activities point of view. And penicillin G and coloxin and vancomycin, bacteriacin and fluoxetine. These all are this particular spectrum of antibiotics is against the narrow range of disease is caused by this particular bacteria. Is fight out and against withstand of that particular the disease caused one, and it destroy. On the other hand, it is inhibits. Yeah, the mode of action wise is in a bacteriostatic and bactericidal. The thing is, it's necessarily it kills them. Yeah, this particular the biological or the chemical composition of this particular, and it stops of this particular the rapidness causing one, it kills them. Yeah, in this bactericidal is also same. These particular agents can work out. This tetracycline, chlorophenicol, erythromycin, and lincomycin. These bactericidal antibiotics comes under penicillin, cephalosporin, erythromycin, immunog- immunog- immunoglutamics, glycosides, and cortimoxazole. All these can work out of their action. Yeah, it suppress or it inhibits this both bacteriostatic and bactericidal. And uses. There all are so many uses of ENT infections and diphtheria and gallbladder infections and skin infections, rashes like fungal infections and gynecological infections, pneumonia and sepsis, abs, septics, and all these are Winston and scarlet fever, whatnot. These all antibiotics can stop and inhibits and controls. Misuse. Once it's misused, then definitely the side effects will causes. Antibiotic. It is called as another hand antibiotic abuse or antibiotic overuse. The misuse it comes under. It may produce a serious effect on the health. It contributes the factor of this certain of the multi-drug resistant bacteria or informal it's called as superbugs. Yeah, it is relatively harmless and at the same time the bacteria can develop the resistance to multiple antibiotics and causes of life and threatening infections. Misuse of this particular antibiotics or overuse leads to superbugs activity. We can identify and overuse. There's a lot of within no time, within a span of hour, so many medicines we may consume, then a lot of effects. Yes, can resistance or cross resistance. In what way? It's continuous usage. When we are concentrate of the drugs requires its inhibits or it can kills the microorganisms. These particular microorganisms is considered to suppress or it can resistant to that particular drug. Yeah, so many examples. We can cross resistance also. Their chemical process, the assay, is similar to the chemical substance. Means the functional groups. It may change, then the resistance of other antibiotics, it results. It gives the results to this particular cross resistance point of view. Possible of side effects. It's a lot of the diarrhea. It leads the bloating, indigestion, abdominal pains, and a lot of the losses of appetite. Sick, being sick, feeling sick, itching, rashes of skin, cough, everything is an allergic, the way of reactions we can identify. The ideal antibiotic, this particular characteristics is maintained. First is simple, readily available in everywhere. That is good antibiotic. It is selective, toxic, and relatively soluble. And this one. Oh, it's a non-toxic. If in case it may exceed, non-toxic. Does not lead to develop antimicrobial resistance. Yeah, if continuously we may use antimicrobial resistance, it may forms that particular antibiotic can forms, not never. It is not that useful. So it is healthy and causing of allergy, or it is in a predisposing the host of their infection. It may leads to allergy or something it may takes, then that is not a good antibiotic character. To identify these particular all these forces through these websites. Okay, we identify how it is acts in antibodies. See, these all antibiotics is in what way it acts in antibodies. You see,
This animation, we will explain what antibodies are, how they are made, and how they are most commonly used in laboratory research. Antibodies are proteins that can specifically recognize foreign invaders or pathogens, such as viruses, bacteria, and parasites. All pathogens express a unique combination of molecules, both inside and outside their cell membranes. These are called antigens. Antibodies can serve two functions.
antigens.
Antibodies can neutralize
Is
pathogens by
opposite
binding to their
one.
antigens and blocking their action, such as preventing a virus from entering a cell or blocking a bacterial toxin. Antibodies also mediate the destruction of pathogens by acting as markers that other cells in the immune system recognize. Antibodies are produced by specialized cells within the immune system called B lymphocytes or B cells. The immune system recognizes a wide array of antigens by producing millions of different types of antibodies, each recognizing a different antigen, with each B cell producing only one particular antibody. Antibodies are Y-shaped proteins and are made up of 4 polypeptides or protein
Remember,
chains,
antibodies is
2
Y-shaped
identical shorter
proteins.
chains called light chains, and 2 identical longer chains called heavy chains. The N-terminus of the 4 polypeptide chains varies significantly in sequence from one antibody to the next and are called variable regions. Variable regions are involved in antigen binding. The other regions of the polypeptide chains vary very little in sequence among different antibodies from the same species and are called constant regions. The constant regions are responsible for interacting with effector proteins that mediate the immune response. Antibodies are produced by B
Light
cells through a
chain, heavy
process of
chains.
recombination, where the genes that code for portions of the antibody protein are recombined or shuffled to create many different variations of the final protein. Antibodies are first expressed on the cell membrane of immature or naive B cells that have not yet encountered antigens. Recognition of the antigen by an antibody leads to B cell activation, which results in its differentiation and clonal expansion. Differentiated B cells or plasma cells in turn produce and secrete large amounts of their respective antibody, which is released into the bloodstream to aid in the finding and targeting of additional pathogens of the same type. Upon antigen binding, B cells can be induced to produce additional antibody variation through a process called somatic hypermutation, where the DNA sequences that code for the antibody's variable regions can rapidly mutate. Antibodies with higher affinity cause further B cell activation, which results in the selection and production of highly specific antibodies to the antigen in question. Because of their ability to bind proteins with exquisite specificity, antibodies are routinely used in research to 1. Quantify the relative amount of protein present in a sample, 2. To detect the addition of a covalent modification and the binding of other proteins to the protein of interest, 3. To isolate a protein or protein complex from a cell lysate, and 4. To visualize proteins, cell structures, and processes under the microscope. To produce antibodies against an antigen of interest, researchers inject the antigen in question or a fragment of it into a mammal such as a rabbit, sheep, or goat. The resulting antibodies are isolated. A polyclonal antibody preparation contains a mixture of heterogeneous antibodies against multiple sites of the antigen. A monoclonal antibody preparation contains an antibody that recognizes only one site on the antigen. Polyclonal and monoclonal antibodies have different strengths and weaknesses. Long-term production of polyclonal antibodies requires the use of multiple individual animals, resulting in differences between different batch preparations. In contrast, monoclonals can be continuously produced from a clonal cell population and are less subject to changes. Since polyclonals can recognize multiple sites within a protein, they can amplify detection signals, but they also then cross-react more with non-specific proteins. Monoclonals, on the other hand, do not amplify detection signals as well, but provide a lower signal-to-noise ratio due to their higher specificity. As a result, polyclonals are better suited for detecting proteins that are expressed at low levels. They are also used to effectively detect different isoforms or versions of a protein or to screen for the presence of the protein of interest in other species. Monoclonals are more commonly used when purifying a protein from a mixture where specificity is important or when detection of a very specific portion of a protein or modification is desired. When detection or isolation of a protein is desired, researchers commonly use a combination of two antibodies. Primary antibodies bind to specific antigens, while secondary antibodies are made to recognize and bind to the constant region of primary antibodies, which is the same within a given species. Depending on the application, secondary antibodies might be conjugated to a marker or a bead, which allows for easy detection or isolation of the protein of interest. The use of a secondary antibody leads to amplification in signal, since multiple secondary antibodies can bind to a primary antibody.
Yeah, this is the story behind antibodies. Through antibody, immunity is develops. Yeah, through antibodies, hybridomo technology. Yeah, antibodies are new technology.
Till now we have studied that in response to an antigen our immune system produces heterogeneous mixture of antibodies. These antibodies are of different specificities, that is, these antibodies recognize different epitopes on the same antigen. The antibodies derived from the multiple clone of B cells are known as polyclonal antibodies. We also understood that polyclonal antibodies have some limitations in diagnostic and therapeutic applications. Today we will study monoclonal antibodies. As the name reflects, monoclonal antibodies are derived from the clones of single activated B cell. So, these antibodies will recognize and bind only one particular epitope on an antigen. We can also say that monoclonal antibodies are identical antibodies with same specificity. The technique of production of monoclonal antibodies was discovered by Georges J.F. Kohler and Cesar Milstein in 1975. This technique is known as hybridoma technology. They were jointly awarded Nobel Prize in Physiology or Medicine in 1984. Let's study in detail the technique of monoclonal antibody production. First step in production of monoclonal antibodies is the immunization of an animal. Usually the animal used is the mouse. Mouse is immunized with the antigen against which we need antibodies.
Yeah, mouse.
Let's say this is our antigen, and it has 4 different epitopes.
Mammal.
Mouse is injected with the antigen several times. As a result, mouse B lymphocytes are stimulated against the epitopes or antigenic determinants of the injected antigen. After several weeks, when these B lymphocytes reach to an optimal amount, the mouse is sacrificed. Spleen of mouse is removed aseptically. It is known to us that spleen is the secondary lymphoid organ, and we can easily harvest activated B lymphocytes from spleen. Spleen is then subjected to mechanical or enzymatic disruption. This results in the release of cells. Activated B cells or plasma cells are separated from the normal spleen B cells by density gradient centrifugation. So, at the end of this step, we have activated B cells which are capable of producing antibodies against the specific epitopes present on the antigen. Next step is cell fusion. B lymphocytes have a short lifespan in cell culture. In this step, activated B lymphocytes are fused with myeloma cells. Here we need to note that these myeloma cells being used are mutated myeloma cells. Myeloma cells are cancerous B cells. They can divide indefinitely in a culture. But their two genes are mutated. First is HGPRT gene. Thus, they are not able to synthesize nucleotides by the salvage pathway. And second gene is the immunoglobulin mutation in these genes. These myeloma cells cannot produce their own antibodies. These mutations are represented as HGPRT negative and Ig negative. Cell fusion is done by mixing two types of cells in the presence of chemical fusogen, polyethylene glycol. As a result of cell fusion, we will have 5 types of cells: unfused B cells and fused B cells, unfused myeloma cells and fused myeloma cells, and hybrid cells, formed by fusion of an activated B cell and a myeloma cell. These hybrid cells are also known as hybridomas. In our illustration, we have 4 types of activated B cells, each specific to one of the 4 epitopes on the antigen. So, hybrid cells will also be of 4 types. Now, our next aim is to select these hybridomas from this mixture of cells. Selection of hybridomas from the mixture of fused and unfused cells is done by using HAT medium. HAT stands for hypoxanthine, aminopterin, and thymidine. Recall that HAT medium is a selection medium from mammalian cell cultures. Selection of cells in this medium is based on the fact that there are 2 pathways of nucleotide synthesis in mammals. Aminopterin present in the HAT medium blocks the de novo pathway. The only way a cell can survive in HAT medium is by using the salvage pathway of nucleotide synthesis. HGPRT is a key enzyme in the salvage pathway. So, if cell has a non-functional HGPRT gene, the cell will die in the HAT medium. This is because for this cell, both pathways of nucleotide synthesis will fail in HAT medium. In this step, the mixture of cells obtained after fusion step is transferred to the HAT medium.
This is a lab class.
Fused and unfused B cells die within few days because of their short lifespan. They are not able to divide indefinitely in cell culture. Fused and unfused myeloma cells also die. This is because myeloma cells are HGPRT negative, and aminopterin present in the HAT medium blocks the de novo pathway. Hybrid cells or hybridomas survive in HAT medium. These hybrid cells are able to synthesize nucleotide by the salvage pathway. The functional HGPRT enzyme is contributed by the activated B. Also, these cells are able to divide indefinitely, and this property is contributed by the myeloma cell partner. Therefore, what remains in HAT medium are the desired hybrid cells producing antibodies against the specific epitopes on an antigen. Now these hybrid cells are again a mixture of B cells, producing antibodies of different specificities. Recall that each of these B cells will produce antibodies specific to different epitopes on the same antigen. Our aim is to select and propagate single antibody-producing hybrid cell. We need to isolate these hybridomas and grow them. Therefore, next step is the isolation of hybridomas of single specificity. This is done by a method known as limiting dilution. In this method, the cells or hybridomas are distributed in multi-well culture plates at very low density. This is done such that, on an average, each well contain a single cell. In the next step, these hybridomas are screened for the secretion of the antibody of desired specificity. This screening is done mostly by two techniques, namely ELISA and RIA. Hybridoma cells producing the desired antibody are identified. They are isolated and cloned in the next step. So, now we have separate clones of activated B cells, each producing antibodies of a single specificity. In each case, the antibodies produced are null antibodies. In the final step, these hybridomas and monoclonal antibodies are characterized and stored. Mostly they are stored in liquid nitrogen. Now these monoclonal antibodies are ready to
Liquid
use in
nitrogen is
treating and
preserved.
diagnosing diseases.
To the ideal one. Choose antibodies developed. Immunization, cell function, selection of head, hybridomas, and isolation of hybridomas in the species. Screening of the products, and cloning, and characterization. Slowly chemical antibodies. Come to the lab session. Last week, the bacterial culture and bacterial growth is over. Antibody. What is antibody? What is T cells? What is H cells? The basic structure of T
In the
cell
last
and
few
H cell.
lectures, we covered cell-mediated immunity, which involves T
T-cell.
lymphocytes. Cell-mediated immunity
Memory
fights
cells.
against intracellular antigens. They kill infected cells, tumor cells, etc. Pathogens, which multiply in the extracellular spaces of the human body. These extracellular spaces are protected by B lymphocytes, which are responsible for humoral immunity. In this video lecture, we will understand role of B lymphocytes and the basic structure of antibody. B lymphocytes originate in bone marrow, and they also complete their maturation in the bone marrow. After the maturation stage, these cells are released into the blood. They keep recirculating between lymph, blood, and secondary lymphoid tissues. When these mature B cells recognize specific antigens, they get activated. This recognition occurs via specific receptors present on B cells, which are known as B cell receptors. Once activated, these B cells proliferate and differentiate into effector B cells and memory B cells. Effector B cells are known as plasma cells, which are antibody-secreting cells.
Plasma cells.
They produce and secrete antibodies specific to the antigen recognized by the B cell. Antibodies are glycoproteins. The basic structure of antibody resembles Y-shaped molecule having two antigen binding sites and a stalk known as Fc region. These antibodies circulate in the lymph and blood. From there, they reach the site of invasion by the
shape of
pathogen.
antibodies is
They
Y-shaped.
bind to the pathogens
short
or
answer
antigens
questions.
and mechanisms that lead to the destruction of the pathogen. Antibodies are also known as immunoglobulins. This is because they belong to a group of glycoproteins known as globulins. The term immune reflects that these glycoproteins play a major role in immunity. The basic structure of all antibodies and the structure of B cell receptors is same. So, B cell receptors are also immunoglobulins. The difference is that B cell receptor is a membrane-bound immunoglobulin, whereas antibodies are secreted immunoglobulins. Let us now study the basic antibody structure. All antibodies have a same core structure. It consists of 4 polypeptide chains. 2 identical heavy chains designated as H chains, and 2 identical light chains designated as L chains. As you can see here, heavy chains are the longer ones and light chains are the shorter ones. The term heavy and light refers to their molecular weights. Heavy chains have more molecular weight than light chains. Now, since these are polypeptides, the N-terminal of this polypeptide chain is present at the tip and C-terminal is present at the base of each polypeptide chain. As you can see in this image, these chains are assembled into a Y-shaped structure. Each light chain is connected to a heavy chain via a disulfide bond. Here, this yellow line is representing a disulfide bond. The heavy chains are connected to each other via 2 disulfide bonds in the mid-region. Besides these disulfide bonds, a number of non-covalent bonds are also
Disulfide bond, heavy chain, and L chain.
The mid-region of antibody has considerable flexibility. This region is known as the hinge region.
Injuries.
The main advantage which this region provides to an antibody molecule is the ability to adjust to different spatial arrangements of antigens or epitopes. In other words, hinge region make possible the rotation and bending of antibody molecule. The stalk of this Y-shaped antibody molecule is the stem region, also known as Fc region. To each heavy chain, short carbohydrate chains are attached. These carbohydrate chains serve many additional functions such as increasing the solubility of immunoglobulins. Before going into further details of antibody structure, let's have a look what we have studied just now. These are the two identical heavy chains and these are two identical light chains. Each light chain is attached to a heavy chain via a disulfide bond. The two heavy chains are linked to each other via two disulfide bonds. Mid-region is known as hinge region and this is the stem or FC region. Short carbohydrate chains are attached to each heavy chain. Here you can see how 3D structure of antibody looks like. Let's study more about light and heavy chains. Light chains. As we said before, these are the two shorter subunits of basic antibody molecule. Each light chain has a molecular weight of about 25 kilodaltons, and each contains about 220 amino acids. In humans,
229
there
amino
are
acids,
two
the
types
very
of light
biggest
chains,
chain.
kappa chain and lambda chain. They are similar in structure and function, but they are coded by different genes. Kappa chains are encoded on chromosome 2 and lambda chains are encoded on chromosome 22. It is important to note here that each antibody molecule produced by a B cell will either have kappa or lambda light chain, but never both. In humans 60% of light chains are kappa and 40% are lambda. Heavy chains. Heavy chains are the longer subunits of the antibody structure. Each heavy chain has a molecular weight of about 50 to 70 kilodaltons. And each heavy chain contains about 440 amino acids. There are 5 types or classes of heavy chains in humans, all encoded on chromosome 14. These 5 classes are designated by lowercase Greek letters, gamma, alpha, mu, delta, and epsilon. They are also written as G, A, M, D, and E, respectively. Each light and heavy chain contain 2 distinct regions: variable regions and constant regions. Variable region refers to the first 110 amino acids of the N-terminal region, in each heavy and light chain. These regions are so called because the amino acid sequences in these regions have great variability. These regions are designated as VL in each light chain and VH in each heavy chain. It is the variable region of a light chain and a heavy chain, which together form the antigen binding site. So, there are 2 antigen binding sites in a core antibody molecule. Further, the variability of amino acid sequences in these regions is localized within certain areas. Since these areas consists of variable amino acid sequences, these areas are known as hypervariable regions. They are also known as complementarity determining regions, abbreviated as CDRs. This is because these regions together form a structure which is complementary to the shape of specific antigen bound by the antibody. Let's say this line is representing first 110 amino acids in the variable region. The CDRs are represented by blue color. Scientists have found that there are 3 CDRs in the variable region of each chain. These are designated as CDR1, CDR2, and CDR3. The intervening sequences between CDRs or hypervariable regions are known as framework residues, these intervening sequences have restricted variability. Now, as you can see here, the CDRs are the regions which actually interact with its specific antigen, or in other words, they form the antigen binding site. These CDRs are brought together when the antibody molecule folds into its native conformation. In this image, the three blue color lines in each light and heavy chain is representing the CDRs. So, now we understand that antibodies can recognize diverse types of antigens because of these hypervariable regions. The region beyond the variable region of both heavy and light chain is known as constant region. They are so called because the amino acid sequence in these regions shows little variation among antibodies. There is a single constant region in each light chain which is designated as CL. Now, recall that light chains are of two types, kappa chain and lambda chain. These chains differ from each other by minor differences in the constant region of light chain. Constant regions in heavy chains vary from 3 to 4. This depends on the antibody class, and it is the constant region of heavy chain which forms the basis of this antibody classification. In a particular class of antibody, all antibodies have almost same constant region, but constant region of one antibody class is different from the another class. The constant regions of the heavy chains are designated as CH1, CH2, CH3, CH4 starting from the end terminal of the chain. Finally, each variable and constant region in an antibody molecule also has at least one disulfide bond. These are internal disulfide bonds.
Okay. Structure. H chain, L chain, and variable chains, Y shape, single bond and double bonds of sulfuric. Some short term carbohydrate features. CH2, CH3, variable chains. All we are writing. Next is enzymes. Yeah, what is enzyme?
Incredible molecules, enzymes, and they're the focus of this video. We'll look at what enzymes are and what they do, their structure, and how they work. Essentially, how the
Enzyme
structure and
structure.
function link together.
What,
There's two theories we'll get into
what
here,
is
the
enzyme?
lock and key and the induced fit.
Lock and key mechanism.
It's pretty cool stuff, and as we don't have any enzymes that can help us speed through this video, let's jump into it. As I said before, enzymes speed up reactions. More specifically, they speed up chemical reactions without being used up in the reaction itself. This means they act as biological catalysts. So an enzyme is a protein that speeds up or catalyzes reactions. Enzymes don't make reactions happen though. That's really important to remember. They can only speed up reactions that already occur. Basically, they're just helpful bystanders. Because they don't get used up in the reaction, enzymes can be reused repeatedly, so a small amount of enzyme can really go a long way. On that note, enzymes catalyze a whole range of reactions. You name one, and it's likely enzymes are involved somehow. They tend to get around. Take digestion, for instance. Starch is used to store energy, but in order to make use of this energy, you need to break starch up. Here's where the enzyme amylase comes in. Amylase is produced by the salivary glands in the mouth, so it's here that it speeds up the breakdown of starch into glucose
Amylase
monosaccharides or maltose
starch
disaccharides.
component.
These are then used as
Amylase
energy.
is starch component.
Because amylase works within the saliva, outside of the cells, we call it an extracellular enzyme. Not all enzymes are like this though. Some work inside the cell and catalyze synthesis rather than breakdown, like the enzymes involved in collagen synthesis, known as hydroxylases. Don't worry, you don't have to remember that name for your exam. In any case, hydroxylases catalyze the buildup of collagen, specifically the addition of groups which allow its helical structure to form. So hydroxylases play a really important structural role. Clearly, enzymes are involved in a range of reactions: metabolic, structural, in breakdown, synthesis, and inside and outside of the cell. They play an indispensable role, and the key to how they work is their structure. So let's get into that. Enzymes are a type of globular protein, meaning they're made up of several polypeptide chains bound together to form a shape. It really varies. Somewhere within this shape is an active site. I tend to think of it as the interactive site, as this is where the enzyme interacts with other molecules. Like all protein structure, the 3D shape of this active site is determined by the enzyme's tertiary structure, and it's so important to get this right. It's incredibly specific. Any substances that interact with the enzyme do so by recognizing and binding to the active site. They're known specifically as substrate molecules, creates an enzyme substrate complex. But honestly, I'm not kidding about the active site specificity. Enzymes will only bind to substrates that are a perfect match. You've got more luck trying to rob a bank than to crack one of these bad boys. So far I've just told you that enzymes speed things up. I haven't explained how they do it. And there are two levels of understanding here. There's what we know for sure, and there's a couple of theories to explain the rest of it. Let's start with what we know. We know enzymes speed reactions up by reducing the amount of energy required to get the reaction going, known as the activation energy. Imagine the activation energy as a hill or a barrier. It must be overcome for the reaction to proceed. So for every reaction, to get from substrate to product, the activation energy must be reached. Enzymes lower this barrier slightly so that less energy is required for the reaction to commence. They lower the barrier by either bringing substrates together in a joining reaction, or by putting strain on substrate bonds within a breakdown reaction. Often, energy is so typically enzymes allow reactions to take place at a lower temperature than usual, such as body temperature. Without enzymes, many reactions will proceed far too slowly to sustain life as we know it. So enzymes don't trigger reactions, but they make it easier for them to happen by lowering activation energy. Right, now onto the stuff we don't know so well. Essentially, how exactly the active site and substrate come together to lower the activation energy. Scientists do have a couple of theories: the lock and key model and the induced fit model. As the name suggests, in this theory, the enzyme is like a lock and the substrate is like a key.
Yeah,
The substrate slots into the enzyme active
substrate
site,
and
like a
enzyme.
key fitting into a lock, and this forms an enzyme-substrate complex. The reaction then commences, and an enzyme product complex is formed. And this is a bit like if you twisted the key and it broke inside the lock. The pieces, or products, are then released from the enzyme, and it remains unchanged and ready to undergo the process all over again. In this theory, the enzyme and substrate are a perfect match. The only problem with this model is that the enzyme doesn't seem to have as rigid a shape as this suggests. It's actually been shown that the enzyme-substrate complex changes shape slightly to complete the fit with the active site. So bye-bye lock and key. In light of this evidence, the induced fit model was developed. In this model, the substrate locks into the active site of the enzyme as before, but this time, the enzyme-substrate complex slightly changes shape to make the fit even better. Think of it like wearing in a new pair of shoes. Your feet only match a certain sized shoe, but even then, with new shoes, you've got to walk in them a little bit to stretch them out before they'll fit just right. It's a similar thing with enzymes. Basically, in the induced fit model, the active site molds itself around the substrate. You still need the right substrate to bind though, so it's still a specific pairing. It's just that the active site slightly changes shape to accommodate the fit even more. And as before, an enzyme product complex is produced and the product is released. Well, clearly enzymes are incredible. We really couldn't live without them. So before I sign off, here's a few key points to take away. Enzymes are catalysts, meaning they
Enzyme
speed up
catalyst.
reactions but don't get used up.
Biological
They do this by
reactions.
lowering the activation energy required to turn a substrate into a product.
Suppress the
Enzymes
activation
have a
energy.
really specific shape which the substrate interacts with, called an active site. Substrates bind to this active site to form the enzyme-substrate complex. The binding of the substrate to the active site is a rigid interaction in the lock and key model, whereas the induced fit model proposes that the active this process. That's it from me. I'll see you in the next video, guys, where we'll delve further into the world of enzymes and start to look at what factors affect their function. Bye for now.
Story behind of enzymes. It's activate the reaction, rate of reaction is decreases, then speed of reaction. Industrial, the way of usage of enzymes in biological, the catalyst of enzymes in industrial products point of view. The bulk product can generate through enzymes.
Biological catalysts which help speed up chemical reactions without getting consumed in the reaction itself. They break down, build up, or convert substrates into new products. Some enzymes present in living organisms include amylase, protease, lipase, and catalase. When it comes to industry, the uses and roles of enzymes vary widely. Some processes aided by enzyme include tanning leather, extracting juice from fruits,
Daily life.
making cheese, baking bread, brewing and distilling beer, and much more. The processes are sped up since enzymes are catalysts of chemical reactions, are very high temperatures to operate. This saves fuel.
Seed germination.
Enzymes help in the germination of seeds. These enzymes become active when more water is present in the seeds. Different enzymes create different products which help the seeds grow and store energy. Amylase breaks down starch into maltose, which provides energy for growth and construction of cell walls. Protease breaks down proteins into amino acids, which make protein for the construction of new cells. Lipase breaks down oils into fatty acids and glycerol to provide energy for the growing plant. Beer is an alcoholic beverage which needs enzymes in its production. Barley seeds are harvested and then allowed to germinate in warm, moist conditions. The amylase enzymes in the barley seeds activate in such conditions, then convert the stored starch in the seeds into a sugar, maltose.
Maltose.
Maltose is used by the yeast in fermentation to produce alcohol in beer. Maltose is used by the yeast in fermentation to produce alcohol in beer. A different process is used to harvest oils from certain plants, such as sunflowers or olives. Oils are stored in the plant seeds for energy, and lipase is present, which would break down these oils into a different product when it is time for the seed to germinate. Thus, sunflower or olive seeds should not be made to germinate if their oils are to be extracted. Pectinase is used to speed up the extraction of juice from fruits. Pectinase is an enzyme which breaks down pectin in plant cell walls, which are molecules that hold plant cell walls together. Pectin is sold commercially as a light brown powder from citrus fruits. This can then be used to make jams and juice. In industrial terms, fermentation is any process that uses microorganisms
This
to
fermentation
produce
is
a
also
beneficial product
one of the
or
topic in
facilitate a
our
useful process.
second unit.
Large stainless steel tank fermenters,
Research.
such as those used for the production of beer, cheese, and antibiotics, provide and maintain conditions fit for the growth and metabolism of microorganisms like bacteria and fungi, which serve as the sources of enzymes. These microorganisms produce enzymes outside of cells to break down substrates such as complex carbohydrates, proteins, and fats, and then digest them. Penicillin is an antibiotic which uses fermentation in order to be produced. It comes from the fungus Penicillium. A scientist named Alexander Fleming discovered its use as an antibiotic in 1928 when he noticed by accident that the fungus was growing on his agar plates, killing off the bacteria around it. Today, penicillin is made in large quantities using industrial fermenters. Fungus Penicillium is put in industrial fermenters. Nutrients such as sugars and ammonium salts are placed in the fermentation vessels along with the fungus. The fungus uses these nutrients to make proteins and nucleic acids. Biological washing powders undergo a similar fermentation process as the production of penicillin. The enzymes released by microorganisms, such as Bacillus bacteria and Aspergillus fungi, are collected from the fermenter through extraction, then used for biological powders and sold commercially to clean clothing. Enzymes break down substrates found in typical stains. Protease breaks down proteins found in blood, grass, and egg. Lipase breaks down fat found in grease stains such as in butter, lipstick, and mayonnaise. Amylase breaks down starch found in food stains. And cellulase breaks down cellulose fibers on cotton textiles to remove attached dirt. In addition, the enzymes in biological powders are modified to let people wash their clothes in low wash temperatures and in alkaline conditions. Newer innovations allow clothes to be washed in temperatures as low as 15 degrees Celsius. These lower temperatures require less energy to operate washing machines. Also, the enzymes and their byproducts do not harm the environment or living organisms, unlike other detergents. These are just a few pros in the long list of advantages that enzymes bring to humans.
Enzymes point. Yeah, this particular, the recent past, one of the student of Vishakhapatnam, they identify this, especially for this dengue fever, malaria, dengue and all other infectious diseases caused by mosquitoes. Recently they identified, you see,
ఎంతో కొంత మేలు చేయాలి, ప్రత్యేకత చాటుకోవాలి. విద్యకాని ఆలోచనలు సాగించిన నలుగురు యువతీ యువకులు ప్రాణాంతక వ్యాధులకు కారణమైన దోమల నివారణకు సరికొత్త ఆవిష్కరణలు చేశారు.
mosquitoes, this.
మలేరియా, డెంగ్యూ వంటి వైరల్ జ్వరాలతో ఏటా లక్షల మంది మరణిస్తున్నారు. ఈ వ్యాధుల వ్యాప్తికి ప్రధాన
మలేరియా,
కారణం
డెంగ్యూ.
దోమల నివారణకు రకరకాల మందులు మార్కెట్లోకి వచ్చిన ఫలితం నామమాత్రమే. ఈ బెడద నుంచి తప్పించేందుకు శాశ్వత ఔషధం కనిపెట్టే దిశగా పరిశోధన ప్రారంభించారు విశాఖ గాయత్రి విద్యా పరిషత్ కెమికల్ ఇంజనీరింగ్ విద్యార్థులు.
Our chemical engineering students.
కార్తీక్, పద్మప్రియ, స్నేహ, శ్రావణి ఈ నలుగురి లక్ష్యం ఒక్కటే, నేర్చుకున్న విద్య సమాజ హితానికి ఉపయోగపడాలి. విశాఖ గాయత్రి విద్యా పరిషత్ కెమికల్ ఇంజనీరింగ్ 4వ సంవత్సరం చదువుతున్న విద్యార్థులు మలేరియా, డెంగ్యూ, వైరల్ జ్వరాలపై దృష్టి సారించారు. ఈ రోగాలకు కారణమైన దోమల నివారణే ధ్యేయంగా విస్తృత పరిశోధనలు చేశారు. పర్యావరణ హితమైన దోమల నివారణ ఔషధాన్ని తయారు చేశారు.
మస్కిటోస్ వల్ల చాలా ప్రాబ్లమ్స్ ఫేస్ చేస్తూ ఉంటారు సొసైటీలో. చూసుకున్నట్లయితే మన మలేరియా, జూకా వైరస్ అని ఇలా ఫీమేల్ మస్కిటోస్ చాలా వాటి వల్ల మనకు ప్రాబ్లమ్స్ వస్తుంటాయి. సో మేము ఏం చేస్తామంటే బీయింగ్ అండ్ ఇంజనీరింగ్ స్టూడెంట్స్ మనం ఏదో ఒక ఇన్నోవేషన్ ట్రై చేయాలి కాబట్టి వీ హ్యావ్ టు డూ సంథింగ్ టు సొసైటీ. మామూలుగా స్కేల్లో చూసుకున్నట్లయితే మస్కిటోస్ వల్ల వచ్చే ప్రాబ్లమ్సే ఎక్కువ ఉంటాయి. మస్కిటోస్ వల్ల చనిపోతున్న మనుషులు ఎక్కువ ఉంటారు మనకి. ఎందుకంటే బికాస్ మస్కిటోస్ కాజెస్ మెనీ డిసీజెస్ దాన్ అదర్ వైల్డ్ యానిమల్స్ టూ. మస్కిటోస్ వల్ల వచ్చేటటువంటి ప్రాబ్లమ్స్ సాల్వ్ చేద్దాం అనేటువంటి స్టెప్ తీసుకొని మేము స్టార్ట్ చేసాం. అది
అధ్యాపకుల సహకారంతో ఏడాది పాటు పరిశోధనలు చేసిన కార్తీక్, పద్మప్రియ, స్నేహ, శ్రావణి గడ్డి మొక్కల్లో దోమలను నిరోధించే లక్షణాలున్నాయని గుర్తించారు. స్టీమ్ డిస్టిలేషన్ ప్రక్రియ ద్వారా నూనె తయారు చేశారు. గ్యాస్ క్రోమెటోగ్రఫీ తదితర విధానాల ద్వారా నూనెకు ఇతర రసాయనాలను జోడించి సరికొత్త దోమల మందును కనిపెట్టారు.
గ్యాస్ అనేది తీసుకొని దాన్ని స్టీమ్ డిస్టిలేషన్ అనే ప్రాసెస్ ద్వారా దాని నుంచి ఆయిల్ ఎక్స్ట్రాక్ట్ చేసాం. తర్వాత ఎక్స్ట్రాక్షన్ అనే ప్రాసెస్ ద్వారా మిగిలిన 5 డిఫరెంట్ ఆయిల్స్ తీసుకొని దాని ద్వారా మేము ఈ క్యాండిల్, ఇది వేపరైజర్ అండ్ ఇంకోటి స్ప్రే అనేది తయారు చేసాం అనమాట. ఈ క్యాండిల్ ఏంటంటే దీంట్లో మేము ఈ ఆయిల్స్ అనేవి పారఫిన్ వ్యాక్స్ అనేది తీసుకొని దాంట్లో మిక్స్ చేసి ఇది సోల్డిఫై చేసాం. సోల్డిఫై చేసిన తర్వాత ఏంటంటే మీరు ఒకవేళ దీన్ని వెలిగించాక దీని నుంచి వచ్చే అరోమా వల్ల ఈ మస్కిటోస్ అనేవి రిపెల్ అవుతాయి ఈ స్మెల్ వల్ల.
ప్రజల అవసరాలకు అనుగుణంగా 3 రకాల ఉత్పత్తులను రూపొందించారు. కొవ్వొత్తి రూపంలో వెలుగును పంచుతూ దోమలను పారదోలే ఉత్పత్తి మొదటిది. ఆల్అవుట్ గుడ్ నైట్ తరహాలో విద్యుత్ ఆధారంగా పనిచేసేది రెండో రకం. మూడోది శరీరానికి నేరుగా స్ప్రే చేసుకునే విధంగా తయారు చేశారు. సామాన్యులకు అందుబాటు ధరలోనే రూపొందించి అభినందనలు అందుకుంటున్నారు.
మార్కెట్లో దొరికే చాలా ప్రోడక్ట్స్లో కెమికల్స్ ఉండటం వల్ల బయట మన ప్రజలకి చాలా ప్రాబ్లమ్స్ జరుగుతున్నాయి. సో దాన్ని మేము ఎక్సీడ్ చేయడానికి మా ప్రోడక్ట్ని ఇన్వెంట్ చేసాం. ఇదేంటంటే మన బయట దొరికే ప్రోడక్ట్స్ లాగా కెమికల్స్ ఏవి మిక్స్ అయి ఉండవు. ఇవన్నీ కూడా ఆరోమా ఆయిల్స్. సో ఈ ఆరోమా ఆయిల్స్ వల్ల మీకు ఏమైనా అదర్ ప్రాబ్లమ్స్ లైక్ హెడేక్, కోల్డ్ లాంటి ప్రాబ్లమ్స్ ఏమైనా ఉంటే ఇది బర్న్ అయిన టైంలో ఆ స్మెల్ వల్ల మీరు రిలాక్స్డ్గా ఫీల్ అవుతారు. యాజ్ వెల్ యాజ్ ఇది మస్కిటో రిపెలెంట్గా పనిచేస్తుంది, యాజ్ వెల్ యాజ్ మీ రూమ్ అంతా ఫ్రెష్గా కూడా అవుతుంది.
దోమల నివారణతో పాటు సుగంధ పరిమళాలను వెదజల్లడం విద్యార్థులు రూపొందించిన ఈ ఔషధం ప్రత్యేకత. ఆహ్లాదకరమైన వాతావరణాన్ని అందించడమే కాకుండా ఎలాంటి దుష్ప్రభావాలు చూపకుండా తగిన జాగ్రత్తలు తీసుకున్నారు. వినూత్న ఆవిష్కరణతో అందరినీ ఆకట్టుకుంటున్న విద్యార్థులు ఈ ఉత్పత్తులపై పేటెంట్ హక్కు కోసం ప్రయత్నిస్తున్నారు. పేటెంట్ హక్కును పొందిన వెంటనే పర్యావరణ హిత దోమల మందును మార్కెట్లోకి తెచ్చేందుకు సన్నాహాలు చేస్తున్నారు.
పేటెంట్ నెంబర్ వచ్చింది, తర్వాత ఫ్యూచర్లో కమర్షియలైజ్ చేద్దాం అనుకుంటున్నాం. మేము ఒక మంచి పార్ట్నర్ కోసం వెయిట్ చేస్తున్నాం. ఆ పార్ట్నర్ ఎలా అంటే ఒక సోషల్ విజన్తో ఉండాలి, అంటే సొసైటీకి ఏదైనా హెల్ప్ చేయాలి అన్నట్టు, నాట్ ఫర్ ప్రాఫిట్ బట్ సొసైటల్ యూజ్ కోసం పనిచేసేటట్టు ఉండాలి. అండ్ ఒకవేళ అలాంటి వాళ్ళు వస్తే వీ వుడ్ లైక్ టు కమర్షియలైజ్. దీని కాస్ట్ చాలా బయట వాటితో పోలిస్తే చాలా తక్కువ ఉంటుంది. ఇది వేపరైజర్ ఒక 2 months వస్తుంది అండ్ కాస్ట్ అబౌట్ 80 రూపీస్. 2 months బయట వాటితో పోలిస్తే చాలా బాగా వస్తుంది. అండ్ క్యాండిల్ ఏమో 8 hours to 10 hours వస్తుంది, కాస్ట్ అబౌట్ 8 రూపీస్. ఒకవేళ ఎలక్ట్రిసిటీ మనకు సపోర్ట్ చేయనప్పుడు మనం క్యాండిల్ వాడుకోవచ్చు.
ఎడ్యుకేషన్లో క్రియేటివిటీ షుడ్ నాట్ బి కిల్డ్. ఆ ఉద్దేశంతో ఇన్నోవేషన్ని మేము చాలా ఎంకరేజ్ చేస్తున్నాం. మనకి నేచర్లో వచ్చే ప్రాబ్లమ్స్కి నేచర్లోనే సొల్యూషన్స్ ఉంటాయి. ఆయుర్వేదం ఈజ్ బేస్డ్ ఆన్ దట్ ఓన్లీ. సిమిలర్లీ మస్కిటో రిపెలెన్సీ మనకు ఆర్టిఫిషియల్గా ఎన్ని గ్యాడ్జెట్స్ ఉన్నా మార్కెట్లో, స్టిల్ వీ కెన్ హ్యావ్ దిస్ బై యూజింగ్ సంథింగ్ ఫ్రమ్ నేచర్. సో అవి ఈ ఆయిల్స్ని ట్రై చేయమని చెప్పినప్పుడు ఈ ఆయిల్స్ని వీళ్ళు చాలా చోట్ల దే హ్యావ్ డన్ ఎక్స్పెరిమెంట్స్ and they have found a mixture which is most effective because they have done in field trials also. Danivalla today they are able to file a patent.
Yeah, one of the mosquito repellent is for one of the chemical engineering background students. Visakhapatnam. Okay, you can leave. I can stop it here. antibodies, enzymes, fermentation, industrial way. All these three you remember. Okay, let's stop it here.